Let's precise relationship between salt concentration and microbial growth.
Introduction. Microbial growth is known to be significantly influenced by various physical and chemical environmental factors. Elevated salt concentrations are observed to increase osmotic pressure with the environment. This increased osmotic pressure leads to cellular plasmolysis and the subsequent inhibition of microbial growth.
Our primary objectives for this experiment were, first, to track and analyze microbial growth under varying salt concentrations to closely understand these cellular physiological responses. Second, to explicitly identify the exact impact that different sodium chloride concentrations have on microbial proliferation over two specific time intervals, 24 hours and 48 hours.
And this is the material we used during the experiment.
For procedure, step 1, 16 tubes divided into groups with 3 replicates, each 5%, 10%, 20% positive control and non-NaCl adjusted nutrient broth. Each tube contained 5 ml of respective media and was labeled accordingly. Step 2, 0.1 ml of bacterial suspension was aseptically inoculated into each tube. Positive control, 3 tubes of non-NaCl adjusted nutrient broth inoculated with bacteria. Negative control, 3 tubes of uninoculated broth used as blank. Step 3. Incubation at 37 Celsius for 24 and 48 hours. Samples homogenized using vortex mixer at medium speed. Visual turbidity compared against uninoculated broth after each time period. In procedure, samples were homogenized using a vortex mixer at medium speed to ensure uniform distribution. Visual turbidity was compared against uninoculated broth after both 24 and 48 hours of incubation. Samples were transferred into cuvette for spectrophotometric analysis at 600 nm, using uninoculated broth as the blank reference.
The data collected after the first 24 hours of incubation was measured via spectrophotometric analysis at 600 nanometers. The 5% sodium chloride environment demonstrated robust microbial growth, reaching an OD 600 of 0.405. At 10% sodium chloride, we observed moderate growth with an absorbance reading of 0.271, showing that the bacteria possess some initial salt tolerance. The 20% sodium chloride environment, however, severely inhibited growth, remaining at a minimal OD 600 of just 0.042. For comparison, our non-sodium chloride adjusted positive control set at a baseline reading of 0.151 at this 24-hour mark. After 48 hours of incubation, we noticed some dramatic shifts in our cultures. The 5% sodium chloride sample continued to climb, reaching an OD 600 of 0.694. The 10% sodium chloride sample also increased steadily to a moderate 0.520. Our non-sodium chloride adjusted positive control experienced the most significant surge, demonstrating the highest overall proliferation with an absorbance of 1.029. Meanwhile, the 20% sodium chloride concentration completely halted any progress, maintaining a severely restricted OD 600 of only 0.037. This directly confirms the powerful impacts of severe osmotic stress on halting microbial multiplication. To summarize our core findings, there are two key observations from this experiment. Observation 1. Microbial growth clearly increased over time within the 5% and 10% sodium chloride environments. This confirms that Staphylococcus aureus exhibits moderate halotolerance at these lower concentrations. Observation 2. Growth becomes severely restricted once you hit a 20% sodium chloride threshold. Ultimately, the non-sodium chloride adjusted positive control supported the absolute highest level of microbial proliferation by the end of 48-hour window, proving that while the bacteria can tolerate lower salt levels, elevated concentrations are highly effective at inhibiting their growth.
We can discuss that the optimal salt concentration for the growth of the tested bacterium was indicated by the non-NaCl adjusted positive control, which exhibited the highest absorbance readings after the 48-hour period. No microbial growth was expected across the negative controls. However, two samples was found to be contaminated due to technical error. Proper sterilization of all components must be performed to mitigate this risk in the future. In accordance with anticipated outcomes, luxurious growth was recorded for both the positive control and the 5% sample. Growth was found to be moderate at 10% and little visible growth at 20%. Moderate microbial growth was supported in the 10% NaCl environment, suggesting that the microorganism possesses a certain degree of halotolerance. In discussion, this severe growth inhibition is attributed to the extreme osmotic pressure created by the hypertonic 20% salt solution, which is known to force water out the bacterial cells, causing plasmolysis. Microbial growth was observed to decrease as the concentration of sodium chloride was increased. These experimental observations are directly related to real-world application, particularly in the field of food safety and spoilage prevention. The traditional practice of salting food for preservation is heavily supported by this finding, as the induced osmotic stress is proven to effectively inhibit the growth of contaminating microorganisms.
In conclusion, our study confirms a clear inverse relationship The more salt in the environment, the slower the bacteria grow. At extreme levels, growth stops completely. This confirms our objective and proves that salt remains a highly effective antimicrobial agent. To conclude, this research is supported by a range of peer-reviewed studies and institutional reports. The full details and URL for these sources are provided here for your review.
Got a friend in me. You got a friend.